Good morning / afternoon to everybody. I am very proud to be introducing our speaker for today's lecture, Dr. Gary Wenk, especially since he has made such a tremendous impact on my personal scientific career. I first met him when I was a student at Ohio State, and he serves as a full professor in the Department of Psychology there. I just remember how his mentorship as a faculty member really kind of fueled my curiosity for studying the brain. So I wanted to invite him here to talk about his work and what he's done over the years. Before joining the community at OSU, Dr. Wenk was able to enrich the scientific community at many other locations throughout the country. Too many to really talk about, but just to mention a few, he was at the University of Cincinnati for his PhD, the Albert Einstein College of Medicine for his postdoctoral fellowship, as well as contributing to the scientific community at Johns Hopkins and University of Arizona. In addition to his laboratory research, Dr. Wenk has written several books highlighting the importance of why we should understand how our brains respond to the non-pharmaceutical components of drugs, such as food and exercise, which I highly recommend. They are fantastic. Largely, his laboratory has been involved in preclinical investigation of various types of drugs and their potential to provide a neuroprotective effect for the neurodegeneration that we associate with normal aging and related pathologies. So to not take up too much time, I will send it over to Dr. Wenk. We're going to hear about a lot of great research today.
Thank you, Lindsay, for the invitation. And I have to point out to your colleagues there that I and many of my colleagues at OSU are very proud of you. You are always one of our stars in the program. So no one's surprised that you've ended up with such a great group of colleagues.
Thank you so much.
We're all excited to watch and see what you do next. So good luck to you. And thanks again for inviting me. I have been studying inflammation in the brain for about 25 years. And when I first started doing this, the reviewers at the NIH kindly reminded me that there is no such thing as inflammation in the brain, that the brain is a protected structure. And in spite of their wisdom, I went ahead and did the research anyway, and began to explore animal models of neuroinflammation, hoping that it might teach me something about Alzheimer's disease. I'm going to skip to the very sort of one of the final points here. Let's go back here. Oh, here we go. Now we're doing it. And say that this is our working hypothesis. Now, when I'm looking at the screen, as everyone knows, I can't see some of the letters because there's pictures here, but I know what it says. I don't know if it's blocked for anyone else. Anyway, our working hypothesis was that chronic brain inflammation does not cause Alzheimer's disease. This is not a model of Alzheimer's. It's a model of a component of Alzheimer's disease that we're using to try and understand what happens in the brain, and then learn from what goes on in the brain to then design, therapies, approaches to reverse the effects. What we discovered was that, and this is sort of a point that I'll come back to, is that inflammation may not cause Alzheimer's disease, but it underlies the regional and neurotransmitter system vulnerability. What I mean by that is I've always been interested in understanding why this neuron in this part of the brain, why acetylcholine neurons, the dopamine neurons, why temporal lobe systems, because as all of you are aware who study Alzheimer's disease or are interested in it, the pathology is distributed in a specific pattern. And what I'm going to show you is that we believe inflammation contributes to that pattern. This is the sort of our model, it's a slide I stole from someone so long ago I've lost the reference here. But many people start over here on the left side of the screen and think, and we know that Alzheimer's is a disease of aging, that there are mutant genes that people inherit, and that things such as head injury and Down syndrome can predispose people to dementia. But we are also very well aware that Alzheimer's disease is caused by lifestyle factors, something that we do every day probably that ages our brain and puts us at risk. And that's where I think up here is where inflammation comes in, and other things as well that I'll touch on these as I go along. But inflammation seems to come in where it impacts neuronal death. And you'll see that inflammation and the consequences of the cytokine proteins and so forth are all capable of killing neurons, and activated microglia are all capable of stripping synapses to impair cognitive function. So you'll see why this happens, I think, as I go along. So our model began back in the actually mid-90s with the help of a French scientist, Beatrice Hausler-Zignac, and you'll see her at the end. We're looking at ways to generate inflammation in the brain of animals. And I started off following some research out of China, which today seems rather ironic. But anyway, we were looking at bacteria, gram-negative bacteria, and we were extracting lipopolysaccharide from it and putting that in osmotic mini pumps, very low doses, and then putting those into the animal's body. These are rats that we're going to be working with. I will point out along the way that we did these same studies in mice, transgenic mice, and if you're interested, I can talk about that, but I'm going to try and keep it focused on rats. And what we found was that when we put the cannula into the fourth ventricle, we put it there so we didn't damage motor cortex, sensory cortex, that the LPS would percolate through the entire brain. You will notice the dose is pretty low. We did try higher doses and it made the animals quite sick. At this dose, 24 hours a day through the mini pump, what we found was that the dose was low enough that the animals didn't actually become sick. In fact, when we would look in their blood, they didn't have elevated levels of serum cytokines or any inflammatory markers at all. And they were quite healthy. They grew, and we actually tested whether or not they would mate normally, and they did. We did studies on female rats. I'll talk about those later as well. But overall, this is the approach that I'm going to sort of center on for the entire talk. It's a slow infusion of a pro-inflammatory molecule, lipopolysaccharide. Now, I did try other molecules such as interleukin, TNF alpha, and those work just as well, but this is LPS turned out to be a lot cheaper. Now, LPS acts by finding toll-4 receptors and activating microglia on them. It's a self-propagating process. Cytokines are produced, TNF alpha feeds back on its receptors, and this system just sort of builds and builds and builds and dumps a lot of cytokines into the environment, which don't live very long, but they live long enough to have an impact on the brain. That's been the model. So let me just show you what happens briefly. Up here on the upper left side is a normal looking brain, and what we're staining for is our activated microglia. And essentially all brains have a little, but you can see infusing sterile cerebral spinal fluid doesn't really produce any inflammation. These we're measuring actually MHC2 class proteins on the surface. At about 2 weeks, what we find is that it starts to show up a little bit more densely. By 4 weeks, you'll notice that it is in the blade of the dentate gyrus, and the concentration is getting quite high. And finally, by 8 weeks, there's an awful lot of activated microglia. But the first thing that jumps out at you here is that even though we're bathing the entire brain in LPS for 8 weeks, we've actually gone up to 150 days by replacing osmotic mini pumps. You can see that the inflammation only is centered on certain parts of the brain. I'll expand on that in a moment. So I can guarantee you that there are a few activated microglia outside the dentate, but for the most part, you can't see them. They're very few, and for whatever reason, only the microglia in some parts of the brain actually become activated. So we went back to that to try and figure out why and how it changed over time. And this is an early study. We started infusing LPS and sacrificing the animals every few days. And as you might expect, after a couple of days, we saw microglia distributed around the entire brain. And each dot there represents about 4 to 5 activated microglia. And simply put, there was activation everywhere in the cerebellum and the caudate and the thalamus, as you would expect. But as time went by and we got to 3 weeks and 4 weeks, the activated microglia in those parts of the brain actually turned off and only remained on in temporal lobe and the hippocampus. So the question becomes why? What is it about neuroinflammation induced by lipopolysaccharide that only seems to turn it on briefly everywhere, and then it goes back to normal? Well, part of that is because we're using young rats. Young brains are very plastic and they recover. But the other part of it is because there's something different about these parts of the brain. Now, I didn't do this study, but a group in Japan actually asked the same question. Do the microglia in the dentate differ from the microglia that are out here in CA3 and CA4? And the answer seems to be no. The reason the microglia in the dentate stay activated week after week after week is because, very simple answer, this sort of architecture. the extracellular space in some parts of the brain is smaller than in other parts of the brain. And they speculated that subtle changes in pH or cytokine or glutamate didn't get cleared as quickly in some parts. And so it just went on and on and on. And you can imagine if this were an older brain, microglia tend to also be unresponsive and don't recover. This process would just continue to go forward. Now, that idea was actually picked up by other scientists, in particular, Eric Ryman out in Arizona. The top slide shows you the distribution of plaques and tangles that are usually seen in Alzheimer's patients. Temporal lobe, parietal lobe are always affected, some parts of the frontal lobe, cingulate gyrus, and you can see also in yellow the basal forebrain acetylcholine neurons, and in green the nigra and Raphe cells are also affected. And what I'm going to show you is that in my model with chronic lipopolysaccharide into the fourth ventricle, it takes on this exact same pattern of the distribution of activated microglia. And when I look at acetylcholine neurons in the nucleus basalis, they begin to die. And the longer the inflammation is there, the more of them die. And I'll get back and show you some pictures of the nigra in a moment. But what Eric Ryman showed was, this is now decades ago, he followed patients and he examined glucose utilization. Siegfried Hoyer had shown in the 70s and 80s that glucose utilization went down in Alzheimer's patients. Eric followed up on this in the 80s and 90s and found out that in the regions where patients would 20, 30 years later deposit plaques and tangles, they were already showing evidence of not just inflammation, but also decreased glucose utilization. The system, the region, those cells in those systems were already being affected decades before the symptomatology showed up. In our animals, because of the inflammation, what we found in our MRI studies of them is that after 42 days of LPS, the ventricles actually started getting bigger. This is not edema. This is actually an increase in size. We tested for that. Now, Leila de Toledo Morel in the 90s and early 2000s had done studies on patients, finding that the same thing happened in early onset MCI patients. There was a volumetric change in the size of the ventricles, and what we found was the same thing, that the temporal lobe itself actually started shrinking. So if it's shrinking, you would predict, as we did, that we may be losing cells, and we actually did stereology to count them. And what we found is that in layers 2 and 3, there was actually a decline in the large neurons. I'm not saying that they died, we just didn't see the larger neurons when we went to count them. But I believe they are actually disappearing, and you'll see why I think that as I go along in a few minutes. So if there is a loss in entorhinal cortex cells, and if there's inflammation in the dentate, it's, I think, an easy thing to predict that there might be a loss of LTP. So in studies that I did with Marina Lynch in Ireland and also Carol Barnes in Arizona, what we found was that chronic neuroinflammation impaired hippocampal LTP. And I think given what you saw with the shrinkage in the hippocampus, the inflammation, it's not too surprising. But the question that this raised, I think, was that are there behavioral impairments that go along with it? And the answer was, yeah, there certainly are. Our animals were just as impaired. So our young, brain-inflamed male rats were as impaired as our old rats in the water maze. And, you know, because, of course, I trained with David Olton for many years, I tried the T maze and the radio maze, and Carol Barnes and I collaborated with the open field maze. And so a number of tasks, and the animals show a standard memory impairment of any animal that has impairments in the temporal lobe. No, I'm only going to be focusing on male rats initially, but I'll get back to the females in just a moment. What we think is happening, and here's my little glutamate dots are showing up, is that from what happens is that with inflammation, inflammatory cytokines, complement, actually interfere with glutamate transporters in astrocytes, and so glutamate levels tend to build up extracellularly. Down here in the lower left, the exchange of glutamate molecules with cysteine to make glutathione actually becomes impaired. That causes oxidative stress as well. Over in the microglia, we know that microglia, when they're activated, will dump out calcium as well, glutamate. And the elevation in glutamate, and the reason we're paying attention to it is because you can see here that's going to overstimulate, that's going to overstimulate the NMDA receptors. So this is where we found ourselves around late 1990s. Was we had a model that was impairing our young animals' performance. Glutamate seemed to be play a role, and when we put glutamate sensors into this region, we found that extracellular glutamate was increasing. So there's got to be a consequence to that I think leads to the memory impairment. So with the help of John Kozlowski and Bruce McNaughton, and in particular Susanna Rossi in San Francisco, we looked at what NMDA glutamate receptor activation might lead to in the area of interest, the dentate gyrus. And at that time, we were focusing on ARC protein. It had only just been discovered. And you're all probably aware of this. I'll take you through the cartoon. Essentially, what happens is that I can get my note, there we go. The glutamate is going to stimulate the opening of NMDA channels. Calcium enters, activates ARC protein. And this is the typical steps that lead up to the production of LTP in a memory. And you can see on the figures, histological figures, that within 5 minutes of the animal learning something in an open field, you can see the two little red alleles there inside the nucleus of the cell. So here's on one gene and here's the other one. Of course, there's always two, and they become unwrapped and unmasked. Within 30 minutes, you can see the mRNA transporting out of the nucleus into the cytoplasm. And then shortly thereafter, there are cells in the dentate that show up as orange. Now, in a typical learning exercise, this is Bruce McNaughton's data, under normal circumstances, when you're learning something, you need to activate dentate gyrus cells, of course, but you, of course, do not want to activate all of them. Bruce called this sparse encoding, and what it means is that in order to form a memory of where you are in a water maze, for example, you don't need to turn on all of your dentate cells. You just need to turn on 2% of them, and that's enough to learn. So here's a model where normal levels of glutamate are stimulating normal amounts of ARC protein in a region that is vulnerable to chronic inflammation. So the next thing we did, this is all Susanna Rossi's work now, and she was a postdoc in my lab, is what we found is you're looking in the artificial CSF. That's what it looks like in the brain of a rat when it's been out wandering around in a new environment. You see that about 2% of the cells are showing ARC positivity. Then we infuse them with LPS for 4 weeks, and you can see what happens. The little blue dots are all activated microglia. And what happened in our animals that we think underlies their memory impairment and the impairment with LTP is that we lost sparse encoding. That now 4% of the dentate cells are just full of ARC, and they shouldn't be. It's too much. You might view it as neural noise. Now, what I've emphasized here in this high power figure is something that we saw quite frequently. What we saw was that there would be an activated microglia cell, that little blue dot over here, and it would be right next to a bunch of neurons that were full of ARC protein. And we speculated that what might be happening is that the microglia are dumping glutamate, and the glutamate is opening NMDA channels and letting ARC just turn on inappropriately in too many places. There's simply too much extracellular glutamate floating around. If that's true, it leads to the prediction that too much glutamate entering through calcium channels should kill those neurons. So the next thing we did was we looked at the prolonged presence of extracellular glutamate in our theory. And what you're looking at here on the left side is artificial CSF infused animals. The red are dentate cells that possess NMDA channels. And the green are just simply non-NMDA expressing cells. And what we found was that in the presence of LPS, the number of NMDA R1 sites actually began to disappear. So the speculation was that, and I can't prove this, is that they might be dying or they're impaired, they're no longer presenting NMDA channels on their surface. But whatever it is, they're not functioning normally. So these changes probably underlie the memory impairment, the LTP impairment. And we think it's all due to this excess of calcium that was going on. So that's kind of where we found ourselves about 15 years ago. And that leads me to essentially some of the topics that I want to touch on here. And I'm not going to cover all of these. I tested probably 20, 30 different compounds for different drug companies. And various spices for people. And thanks to Jim Joseph in Boston, blueberry extracts and caffeine, because who doesn't want to test that? And insulin, it went on, and estrogen. I'm going to touch on a few of these and just mention them as some of the others as I go past them. So the first thing I did, of course, since I've been collaborating with the scientists at Merits in Germany for many, many years, is look at memantine. Now, memantine was approved for treating Alzheimer's patients. It doesn't work that well, but it has a very neuroprotective, I think, proclivity, and that's what I decided to take advantage of. It is a good tool. It blocks excessive calcium entry. And so what we decided to do is give it to our animals. And here you're looking at the top figure there again of an LPS treated animal. And what we found was that excessive calcium ion entry via NMDA channels could be blocked by memantine. Now, keep in mind, we're giving the memantine every day that they're getting LPS. So there's never an opportunity for the LPS to build up and increase the glutamate. If it did, the memantine was there to block it from, we think, overexciting the NMDA channels. What surprised us was that memantine actually normalized the ARC expression. That's the lower figure, LPS plus memantine. It went back to 2% in all of the cells, and the animal's performance went back to normal. But what really surprised us was that memantine also reduced the number of activated microglia. We didn't expect to see that. We figured it would be still activated, but not actually having a consequence on the dentate cells. I don't have a good explanation for why microglia turned off in the presence of memantine. Microglia express NMDA channels. They express many receptor channels. And maybe the memantine was binding there. I don't actually know. We didn't get that far in the research. The student left the lab. So the other thing that happened in our memantine animals that we decided to look at is neuronal death. I want to point out again that when we infused our lipose polysaccharide, the infusion happens right near the substantia nigra. And as many of you are aware, substantia nigra neurons are vulnerable to inflammation. They are vulnerable in Alzheimer's disease. And we decided to look at our animals. And what you can see in the upper left-hand corner is a control, the artificial CSF plus saline. And when we gave them inflammation, now keep in mind, the inflammation is being deposited right above these cells. You can see the red cells are the microglia, and those are quantified by the chart below on the left. And when we started treating them with memantine every day, we actually were able to prevent the loss of dopaminergic neurons. So neurons that are vulnerable to inflammation, we think die due to excessive glutamated NMDA channels, and we think the memantine data shows that that is the likely mechanism. And anything you can do to reduce glutamate in the synapse or reduce glutamate's action through NMDA channels, you can rescue neurons. But what does this mean? Yeah, we have more Th positive cells, but we can't wait until the cells are dying and then give people memantine. This kind of data implies memantine something they should have in their, their breakfast cereal, taking every morning, just in case, but we do know that Parkinson's disease is most common in the US in people who grow up in rural environments. So maybe individuals who grow up in rural environments may want to consider this kind of medication through a large part of their life, but it's speculation, but it was an impressive protection that we saw with memantine. The next thing I did, of course, is look at antioxidants, and I tested many of these for drug companies. We looked at aspirin, ibuprofen, and others. And if you've ever worked with anti-inflammatories in animals, these things, just like with humans, are very hard on the gastrointestinal system. And when you give it with the diet, you never know how much the animal is getting, and I wanted to inject them. So I began collaborating with a group, Nicox, in a French company, and looking at some of their compounds. And I want to tell you about one of these, just one compound, Nitro-Flurbiprofen. Okay, first of all, flurbiprofen is just like ibuprofen. It's sold in Europe. It's more lipid soluble. I'll show you in a moment. And we decided to look at young rats and male rats in this study. And having said that, let me just reiterate something that I mentioned earlier. We tested old rats in every one of these studies with every drug every time. And I'll come back to this, but every drug fails in an old rat. It is very easy to reduce inflammation in a young rat. You can do it with insulin, you can do it with caffeine, you can do it with many things, many herbals. Everything works in a young rat. Nothing ever worked in an old rat, and nothing ever did except one compound. We also looked at male rats in this study. Now, I mentioned to you, I had a student, Lisa Marriott, who's now a professor in Washington State, and she looked at female rats. And what we found was that you can put LPS in the brain of a cycling female with intact ovaries and completely infuse the brain for weeks and weeks and induce activated microglia. And the females couldn't care less. Their performance is perfectly normal until you take their ovaries out. If you take the ovaries out, they're as impaired as the male rats. So having cycling ovaries is an incredibly protective thing from inflammation, which does make a prediction. We tested it. What happens if you give estrogen back to an inflamed female overactimized rat? What happens is that it does not rescue them, makes them much dumber, much faster. And indeed, about the time we published that study, a group led by Molnard discovered that giving postmenopausal women in the early phases of Alzheimer's disease estrogen, chronic estrogen therapy, actually accelerated their dementia. So we saw the same thing in our young females. So we ended up focusing on male rats because they're just vulnerable to inflammation. Now, a colleague of mine in Italy actually did transplant ovaries into male rats with inflammation, and ovaries in a male rat actually are neuroprotective. So I guess what that means is that, you know, if you're a young woman, never go through menopause. It just isn't worth it. So what we decided to do was test this compound, Nitro-Flurbiprofen. This is what it looks like. Ibuprofen looks just like it, except for that ring on the left and the fluorine. Why did we go with this? Well, the company Nicox invented a whole bunch of these NO-releasing anti-inflammatories. So what happens, as you can see here, it meant I can give animals very high doses and the nitric oxide molecule will fall off, vasodilate the vessels in the gut, and prevents gastritis. And what we found is by doing this, we could make drugs more accessible to the brain. And we could feed it to them, and it wouldn't cause any gastritis or GI impairments. And it made the drugs more potent, as you're going to see as I go along. So Nitro-Flurbiprofen then, with LPS, was pretty good. It cut the inflammation by way more than half. It's not perfect. I'll show you perfect a little bit later, but Nitro-Flurbiprofen was a good compound. It was safe for the animal's GI, and it crossed the blood-brain barrier. In the water maze, you can see here on the left, the first one is CSF. The animals who got CSF got better. Animals who had LPS were impaired, as they always had been. These are young rats with inflammation. And Nitro-Flurbiprofen was completely neuroprotective in terms of reducing inflammation and also rescuing their performance. Okay, I'm going to stick with Nicox's compound for one more time. Many of you are familiar with the work of Sally Frosty, Greg Cole, and others who have been looking at curcumin. Curcumin's a fascinating spice. I looked at other spices, but this one was really quite interesting. This is what we did. Curcumin is an anti-inflammatory spice, very popular in India, and epidemiological studies had shown that individuals, as they got older in India, tended to not develop Alzheimer's at the same rate as the rest of the world, and many of the explanations have centered on curcumin. You can buy curcumin now on the internet for a variety of reasons. But what curcumin does in the gut, as you can see in my figure, is that it will spontaneously split in two at pH 7.2 at body temperature, and it forms the actual active compound, ferulic acid. Now, ferulic acid is an acid. I mean, look at that carboxylic group. It's irritating to the gut. So you want to eat it with food. Now, most people do. Now, on the lower left, you can see LPS. In the middle, ferulic acid was nicely anti-inflammatory, not bad. But when we attached an NO molecule, thanks to Nicox scientists, it was even better. It was even better because even though we're giving them the same number of molecules, it wasn't as irritating to the gut as ferulic acid was. And so it was absorbed faster. So it just simply more got to the brain. I don't think it's any more effective. It's just a bigger dose, more effective levels inside the brain. So does curcumin work? It does. You have to use it every day, and you need something with food, and that's probably just fine. But like many compounds, these kinds of anti-inflammatories, antioxidants, they wear off after a day, and you need to keep using them. And that's true of all the compounds that I tested. We had to give them every day. But the nitric oxide was an interesting connection, molecule connection to be neuroprotective and also GI protective. And we never got back to that. These compounds are not available in the market. Most of them never will be. But Bayer Labs did buy NO aspirin, a nitric oxide relieving aspirin, and we'll be releasing it soon. And it will simply overtake aspirin because it would be so safe and no one will get GI distress. And I'm sure that's coming in the market soon. Okay, I'm skipping blueberry extracts, caffeine, and insulin and getting to something. This was what I was working on the final 8-10 years of that my lab was busy. And thanks to Yannick Marchalant, you'll see in a moment, we decided to go in the direction of cannabis because everything I tested wasn't getting into the brain very well without an NO molecule attached to it. It was hard to control, which compounds got across the blood-brain barrier at a high enough level. And we decided, well, cannabis is something that crosses easily. And there was a small literature out there of people using cannabis to treat the inflammation in individuals with multiple sclerosis. But this is how cannabis works. The CV cannabinoids, in this case, anandamide or 2-AG, there are actually 7 known different cannabinoids in the brain now. But essentially, what it does is it's released postsynaptically, and it goes presynaptically, binds to cannabinoid receptors, G proteins, very common in the brain. And it leads to the blockage of calcium channels and hyperpolarization of potassium channels. And very simply put, cannabis prevents the release of almost any transmitter that requires calcium. So there's no specificity here, none. But it does block the release of glutamate. So at the very least, I was going to find out whether or not it might help in our animal model because it gets across the blood-brain barrier. So that's what we decided to do. And as I said before, we always use male rats in our studies. And of course, in this studies, we also use male rats. And this is what we decided to do. Now, I played around with just cannabis, but we decided, well, that doesn't sound right. But anyway, we did test cannabis in different forms. And we ended up using an analog of cannabis, an agonist, Wenk compound, which had been invented 20 years ago. And it's a CB1, CB2 agonist, and it also blocks TRPV1 receptors. So the entire cannabinoid system, extended system, is affected by it. And it was also very stable. So this is what we're doing. We're putting LPS into the fourth ventricle again, right? And it's young and old rats, no LPS in the old rats, because what we found was we can't make it any worse. LPS doesn't make an old rat more inflamed. They're already pretty inflamed as they are. And that's what we're looking at. Male rats, young and old, and the young get LPS, and we put them in a water maze. And what we found surprised us. First of all, this is the young rat data, all right? On top are the controls. And 0.5 milligram per kilogram and 1 milligram per kilogram was the dose that we were using. Why did we choose those? We had heard from the literature, in the multiple sclerosis literature, that 1 to 2 puffs a day was enough. And we scaled that down to what a rat might take, weighing, 400 grams. And this is the equivalent, we think, of a puff a day. And on the bottom are the LPS animals. Here's LPS in the dentate, 0.5 milligram per kilogram. You can see it actually worked pretty well, as good as Nitro-Flurbiprofen. And when we took the dose still higher, there was even a bit more benefit, but we're really surprised. This is in the next slide. What we found was that the native existing inflammation in the hippocampus was significantly reduced by essentially 1 puff a day of cannabis in the old rats. And we were shocked. This is the first time that any compound had any effect on our old rats. Not only that, but we put them in the water maze. What we found was that the old rats' impairment actually dissipated. They improved almost back to the performance level of the young controls. So we were astonished by this. This is just the equivalent of a puff a day for 3 weeks, all right? But it went further than that. We decided to look at neurogenesis. There's a lot of backstory here, but the scientist Yannick Marchalant had reasons to examine it. And what you're looking at on top in the white and also the fluorescent green these are cells undergoing neurogenesis in the dentate gyrus. And young brains undergo a lot of neurogenesis, right? Between 700 to 1,000 new neurons a day is the estimates. Different estimates, but if you look at B, that's an aged control. That's there's no neurogenesis going on there almost at all. What surprised us was that after only a few weeks of this puff-a-day of the Wenk compound, cannabinoid agonist, the neurogenesis started to come back to normal. Not normal, but just started to come back. And that happened coincident with the animals' performance getting better in the water maze. Okay, so if you look then up close, this is what the young rat looks like with neurogenesis, and this on the lower right you can see what happens in our aged rat. So what we wanted to do, we never got back to it, was to find out how far we could push this and then somehow inhibit it to find out what was going on. Well, we couldn't get to lower doses. It didn't have enough time. Ultimately, the student working on it left. But what we did find was in a series of studies that if we selectively blocked CB1, CB2, or the TRPV1 receptors, any one of those was capable of undermining the neurogenesis effect. So the Wenk compound activated neurogenesis, and blocking CB1, CB2, or TRPV1 were all capable of reversing it. So we really wanted to get back to this. We didn't have time, but it made us wonder what was going on in terms of in the brain with the cannabinoid system and who was responsible. So this is our model that we're working with. We're speculating that the NMDA receptor and is affected by amyloid and inflammation and oxidative stress. That leads to failure of the mitochondria. And the reason I'm putting it there is Henryk Wisniewski long ago told me that he always thought that Alzheimer's disease always began in the mitochondria. And I think he's right to a degree, because that's where inflammation is affecting it. The loss of mitochondrial function leads to the failure, or at least the weakening of the resting membrane potential, and the loss of the magnesium ion block. And that leads to too much glutamate binding, too much calcium coming in, overstimulation of ARC, and then death of the neuron. And cannabinoids are able to prevent the release of glutamate due to the inflammation. That's the model that we were working with. So we decided to push this a little bit and ask, is there anything else going on in cannabis that might be doing this? And what we found was that even CBD, which has a modest anti-inflammatory effect, but CBD primarily acts at adenosine receptors. That's how it seems to produce the relaxing effects and so forth. And so what we found was that CBD could reduce TNF-alpha cytokine levels. And also, and if you looked at adenosine knockout mice, the CBD didn't work. So we think that CBD, as part of a typical, cannabis plant, has its own benefits in inflammation. And so it argues that maybe somebody needs to enhance cannabis in general. All right, and we tried to do that. So the last two studies here. There's a lot of talk in the streets about people who use marijuana using Tylenol to enhance the cannabis. We decided to follow up on that. Turns out that acetaminophen is converted into paracetamol, which is converted into AM404, which looks just like an anandamide, the endogenous cannabinoid. And not only that, but that both of those are potent inhibitors of cannabinoid reuptake. So this is like the cannabinoid version of an SSRI. So we got a hold of a cannabinoid reuptake inhibitor. It's called UCM707. And we gave it to our aged rats because, who cares about the young guys, because everything helps them. So this is an old rat brain, and treated for 3 weeks with UCM707, which is a selective reuptake inhibitor of anandamide. And it worked beautifully to reduce inflammation. And I'll put in another sort of result. We did test FA inhibitors, and they were ineffective in our model. So for whatever reason, FA doesn't play a big role in whatever process I'm dealing with here. So finally then, what we concluded is it appears that selectively augmenting endogenous cannabinoid function is sufficient to achieve all the same benefits that you can get from the plant. And when you think about it, which would elderly patients prefer to do? Inhale a burning leaf or take a small white pill. And a number of the companies that I have worked with the past 10 years are inventing novel cannabinoid enhancing medications that are not in the plant that they can patent, that will act like UCM707 to enhance background levels of cannabinoids. And you can also see this might be very effective as a pain reliever as well. Okay, so I'll try and conclude here so we leave about 15 minutes for questions. But the cannabis, we think the cannabis receptor stimulation then presynaptically is an effective way to reduce the inflammation, to prevent the glutamate release, and to undermine what the inflammation is doing. We think it reduces inflammation, and by doing that, it's able to restart neurogenesis. The reason that matters, I think, is that stem cells that are undergoing neurogenesis have cannabinoid receptors on them. That's why it's so very dangerous for young people to use cannabinoids. It interferes with normal development. But apparently, from our studies, aged stem cells still express cannabinoid receptors, and those can be targeted by simple reuptake inhibitors. Now, we do not know if compounds like UCM707 will make a person high. That could interfere with their effectiveness. Elderly people don't appreciate feeling high, especially patients who are showing early signs of dementia. But we're thinking that that might be in combination with other drugs, such as memantine or herbals or spices or nitro-inflammatories together might be able to reduce what's going on at this glutamate synapse. And I think restore neurogenesis, restore LTP, restore learning and memory. So let me say thanks to the people. These are students who are at University of Arizona and also Ohio State University. They all went on to get their PhDs. And of course, thank my mentor, Peter Davies, who died last year. He was instrumental in helping me get started and looking at acetylcholine. Beatrice Alsford-Zinyak, there she is doing surgery on a rat. She brought the LPS model to my lab, and Susanna Rossi, who's now at UC San Francisco, conducted the studies on sparse encoding and ARC. And so with that, let me stop there and see if anyone has questions. Thank you for your attention.